So thank you and welcome to our webinar. This is a joint presentation by the International IVF Initiative and the New England Fertility Society. This is our third quarterly meeting, and we usually start our meetings with a cocktail party. So this is the cocktail portion of our meeting. Drink fast. NEFS is an inclusive, voluntary, nonprofit organization providing continuing education to infertility professionals in and around the New England area. It was founded in 1988 by Dr. Selwyn Oskowitz. We host 4 meetings per year. 3 are dinner meetings in the Boston, Massachusetts area, and the 4th is a 2-day annual meeting. I would like to invite everyone to our meeting next spring in Providence, Rhode Island, with the theme of Life's a Beach, hopefully in person. Please check out our website at www.nefs.org. We are currently accepting submissions for the Fairing Sponsor's first and second year MD Fellowship Grant. Fairing, through NEFS, sponsors 2 $10,000 grants for research that is open to our members. The winning fellows have 1 year to complete the work and then present their findings at our annual meeting. The deadline for submission is October 1st, and the winner will be announced at our next webinar in December. Now I'd like to turn it over to Paula Dwan, New England Fertility's current president.
Thank you, Evelyn. Welcome to the 34th session of the International IVF Initiative, or I3, and the second virtual New England Fertility Society, or NEFS, meeting. Hello, everyone, and welcome. I3 is honored to help us provide the reproductive community during these unprecedented times with a platform for continued education and collaboration to help you stay engaged, productive, and in touch with others in the IVF community. Worldwide, through our webinars and online materials and an array of relevant topics. We're so happy to have all of you join us. It's an honor and privilege for New England Fertility Society to partner with I3. We'd like to encourage all professional organizations, if you haven't already done so, to join this initiative as other local, national, and international groups have done. As a reminder, I3 is a neutral organization with the prime mission to serve you, the reproductive community. So please help inform I3 of the mission of the topics you are interested in and how they can be of help. All of the I3 webinars are recorded and posted on their website if you wish to revisit them or if you missed any of the previous sessions. I'd also like to just thank the NEFS sponsors. So our sponsors for 2020 at the Emerald level, Semaphore. Our corporate sponsors, Freedom Fertility Pharmacy, Merck, Ferring Pharmaceuticals, New England Surrogacy, and Village Fertility Pharmacy. And our sponsors at the Friends level, Embryotech, Fujifilm, Irvine Scientific, and Seattle Sperm Bank. You can also visit the New England Fertility Society website, www.nefs.org, to see our virtual exhibit hall to visit the sponsor sites. This evening, we encourage all of you to use the question and answer function to ask questions. We have 2 speakers. We would like to ask you to avoid using the chat feature for questions, as our panelists will not be monitoring the chat. We will try to address as many questions as possible. We will have a question and answer session after each individual speaker, and then a combined question and answer session at the end of the program. This evening, Kathy Go, the New England Society Vice President, will introduce our speakers. Kathy Go and Evelyn Neuber will moderate the question and answer sessions.
We're pleased to welcome Dr. Zaninovic to our seminar this evening. He received both his master's and doctoral degrees in biology from the University of Zagreb in Croatia. He joined the Weill Cornell Embryology Laboratory in 1995 and became the laboratory director in 2010. Dr. Zaninovic is also the manager and primary investigator at the Human Embryonic Stem Cell Derivation Lab, where he has derived 43 stem cell lines. What we're looking forward to this evening is listening to his research on embryo selection using time-lapse photography and the use of artificial intelligence in reproductive technologies. Dr. Zaninovic, welcome and thank you for your talk.
Thank you very much for introduction, Dr. Go. Of course, I want to thanks to the organizers, New England Fertility Society, as well as International IVF Initiative. So I will be talking about artificial intelligence, current use of that technology in ART or specifically in IVF protocols, and as well as what can be looking for the future. Okay, so the learning objectives will be, I will briefly go through the principles or concepts of the machine learning or artificial intelligence and how this can be applied. I also want to touch base on what is done so far and what kind of capabilities of this artificial intelligence has in the context of ART. And of course, I want to touch base on the future projects, what we are working on. I am involved in the many different AI projects and many different AI companies, but for this particular lecture presentation, I have no disclosures. So I will start with the current IVF treatments. And we know that we are lacking the standardization in all the aspects of the IVF that we do. And we're lacking the aspects from the laboratory point of view where we actually do not agree, and not in general, during the embryo evaluation and selection in many times. But also we have a lacking of standardization on other parts of the IVF, like for example, diagnosis, treatment, stimulation, and so on. So what is the future that we are looking for? How are we gonna make this better? Well, I will argue that one of the way to do it is that we need to first do personalized treatment of IVF. And secondary, we also have to do precision treatment, because ultimately everything is about time to pregnancy, and we wanna also make time to healthy baby. So a little bit about personalized precision medicine, I will call. Well, precision means that we really have directing our treatments to the maximum results. So it's very precise treatments. Personalized means that each patient is unique, and we cannot just use a general treatment plans for all of the patients. So we really have to tailor or create programs, algorithms, or whatever treatments you want to call it, that will be specifically treated for each particular patient. So for example, if we have a group of patients, they're all different, and we do a therapy, and that therapy is given to all of the patients. Some patients will benefit, some patients will not, and even some of them will be adverse effect. So this is just like one treatment for everybody. But with the personalized medicine, you really are trying to use any diagnostic markers that it's tailoring each of the treatment therapies to each particular type of patients. So this is a theory. How we can get there? Well, of course, artificial intelligence or machine learning should get us there, at least faster. So what is AI? It's a basically partnership between the man and the machine. Machine means a computer. And how good this partnership will be, it's really how well we're going to use these computer programs, how well we're going to teach the computers what to look for. So this is the challenge, how well you do this and what kind of outcome or what kind of results you're looking for. Now, AI was known for many, many years. However, rebirth of AI, I will say, it started in the recent maybe 10 years when we start having very powerful computers, and that we can really train these computers in networks that will perform on the big data. Because we need to have a computerized data. We cannot use pen and paper anymore. So we need to have all our data digitalized. And there are now thousands and thousands of data that is digitalized. The question is, how can we use it? So humans cannot use, maybe can absorb maybe thousands of data points, but the computers can do billions. And of course, the other thing is the really development of the neural networks or computer programs that can allow us to really use this technology. So we know that AI technology is all around us. We're having every aspect on our daily life is actually approached by AI, or we are using it, not even maybe knowing. So the question is, how can we use the technology? How can we, in what manner and what capacity in our daily life in the work? So types of AI, everything is about what we call machine learning. It's really emerging between computer science and mathematics. And depends on what you are trying to do. What is your results outcome you want to have? Either efficiency, classification, or prediction. You are using different type of machine learning concepts or algorithms that you're going to get the results you want. Ultimately, everything of that will actually help us to do better, faster, and more accurately. On the end, everything is really going toward automatization. So briefly, what is AI and how that really works? It's actually, idea comes from our neurons. If we know how neurons work and how all these connections between the neurons is really happening, we will actually do very well in the computer world. So basically, you will have input layer and you have output layer. Input layer is what you are learning or training your computer. So these training data are very important because what type of data you have, the data are balanced, and what diversity of that data will determine how efficient your output layer or results will be. And you can use different type of input layer, like you can use images, you can do videos, you can do numerical data, and then results depends what you want to do. You want to identify something or you want to predict something. Now, what is between? Between is what we call black box, because we don't really know how computer connect these things. So it's really kind of unknown what is the black box. Now, a lot of times you're going to hear about convolutional neural network. What does it mean? It's just what we call deep neural network. So it's basically adding another dimension to the neural network, which adding dimension of depth or 3D. So it's just more complex network that you can really try to find the best results. So AI application in medicine already happened. There are thousands of examples in medicine that it's already happening. One is reading handwritten or electronic medical records. There are actually programs that are using pathology images, and AI can actually, in the many studies, recognize tumors in the pathology images better than actually pathologists. And everything is kind of going towards digital MD. How are we going to get there? How are we going to use this? It's a big question. Now, recently, it was recognized that actually the future of the babies might be applying AI. Now, this is, of course, for the debater. And there is actually another way of looking at it, like if you have a failed IVF, what else can you do to help the couple? And can AI algorithms help us to get there? So I will focus mostly on the AI in the embryology laboratory. However, I will also touch base, what else can we do in all aspects of IVF? In embryology laboratory, we can really use AI for many different things. The whole thing, it's about how to make it efficient and how to make standardized. So you can really use it, AI, for QC and QA. We can use it for sperm evaluation and identification, oocytes or embryo evaluation and prediction. We might be able to use it for the ploidy, non-invasive ploidy prediction and implantation. Now, of course, we can use all that as well on the clinical aspects. For example, I would argue, and we are working on many different aspects of it, how we can use this to evaluate ovarian reserves, how we can evaluate optimal stimulation protocols, uterine evaluation or receptivity. You can use also donor matching and recipients. So there are many different aspects that we can really use this for. So it really depends what your input layer is, what your output layer is, and you can use different methods of different labels or inputs. You can use static images of the embryos. You can do time-lapse videos. You can use time-lapse morphokinetic points. You can do patient demographics and many different other things that we can include. Everything it's about what really you want to achieve and how we're going to get there. So in clinical embryology, I will look into a couple of different things that we can use AI. One is automatic annotations if you are doing time-lapse. Second is evaluation of the normal divisions and quality of the embryo. Third will be blastocyst evaluation or grading and how to choose the best embryo or the most optimal embryo. Now, all these things we can use to predict implantation, predict ploidy, even maybe predict miscarriages. So one of the examples what we are working on, and this is done by Dr. Jonas Malmsten in our group, is basically how to use AI to automated cell division detection, which means how to automate T times. And he was able to use time-lapse of human embryos, and he was able to achieve up to 5 frames, accuracy of 93.9%, which means that he can really do automatic annotations, what currently we are doing manually. And we know that this will extremely help us to standardize the whole process of evaluating the embryos. He's not the only one who is trying to do this. There are many other groups who are also trying to get to the similar methods. The other question is, can we automate the grading of the blastocyst? Because most of the people are using Gardner system of the blastocyst grading. And of course, the grading, it has problems. People have a different way of grading them, and they are not very concordant. So one way of doing it is basically you're trying to use AI methods that you can separate or segment ICM versus trophectoderm versus, let's say, zona. So by doing this, you might be able to actually create automatic grading system. And this is actually exactly what many other groups are trying to do as well. One of the pioneer papers that was published actually from our group is to see how you can use AI in really assessing the blastocyst and assessing the quality of the blastocyst. And if you look at on the top left corner, you can see that even if you have 5 different embryologists and you give them the same image of the blastocyst, that image will be classified differently. Some of them will be excellent quality, some of them will be mediocre or even poor quality. So the whole concept of standardization is very difficult. So by AI, we can basically standardize the blastocyst classification, and we basically can say, okay, this is a good quality blastocyst, this is poor quality blastocyst. And we were able to do this up to 97% of the time. Now we can use this method in many different ways. You can actually combine this method with, let's say, age, and you can actually predict accurately what will be each embryo implantation potential. But the other question is, what is AI really looking inside of the embryo? And actually, by using class activating mapping, or called CAN, we are looking into time-lapse images of embryos that are stork positive, which means good embryo, versus stork negative, which is not good blastocyst. So 2 things we can see. First, AI is really looking within the embryo. That's a good point. The secondary, that AI is really looking on different parts of the embryo duration of the time lapse. Maybe it's looking just in ICM, maybe it's not. So it's very interesting, and we need more studies to figure out where is AI really looking at. We were able to actually create an interface that we can easily upload the images, and this is created by single image on 110 hours, and we actually Stork, with our AI program is called Stork, will give us the prediction of good quality embryo versus bad quality embryo. I will challenge the audience, including myself. I think that our whole process of IVF is on the wrong feet. I think we are having our selection on the end of the process. We are selecting embryos. We are doing very little about selecting the gametes. And we are wasting a lot of embryos, and we like it or not. We are creating form of life, and we shouldn't be doing this without being very respectful to it. So I will argue that we need to create AI algorithms or any other tools that can really give us the selection of the gametes versus selection of the embryos. And one way to do it is, for example, when we have a testis sperm, it's going to be extremely helpful if you have AI who can recognize the sperm in this mess of the testicular tissue, and then we can really kind of easy make out our lives. Here's the question: can AI predict euploidy of the embryo? I can flatly say 100% of the time, no. However, not even PGT biopsy will give you 100% of the time. So let's think about that. So the whole idea is, and there's a lot of efforts to create non-invasive methods of predicting of embryoploidy. And one of that methods will be AI, possibly. And what we present this year on ASHRAE, we are trying to understand, when we look at the distribution of ploidies versus age, can we create a model that could recognize majority of the embryos be euploid or majority of embryos be aneuploid? What is going to give us? It's going to give us the opportunity to say, okay, this embryo, it's worth biopsying, and this embryo is not worth biopsying, because it's either abnormal or highly abnormal or it's normal. So I think this is another approach, not to predict exactly what the ploidy will be, but predicting high accuracy to do invasive methods or not. We go even further. We are presenting this year on ASRM, where we actually create the AI model that will give us exactly prediction for each of the embryo. And we are using different inputs. We are using image of the embryo, we are using age, we are using blastocyst grade, morphokinetics, and we do this all together. So try to see, and we are actually reaching 70% accuracy, which I think is pretty decent. There is actually a recent paper published from a group from Mexico with some other collaborators, which are trying to do similar things. They are using AI algorithm, ERICA, to predict ploidy. I will applaud for the effort, and their accuracy is 0.7, which is great. There is some issues with that. Of course, we have a high false aneuploidy rate. I think it's related to the number of cases that they used. But I think this is way to go towards the end of the not using invasive methods. Can AI predict implantation? Yes, of course, these are the many papers are trying to do the same. I will argue that you should not try to predict live birth, because we know that it's not everything about embryo. Without adding other clinical parameters of uterus, metabolomics, or any other parameters that are important for the implantation, we cannot really go towards predicting live births, but we can maybe go towards fetal hearts. There are also attempts to predict miscarriages. We presented last year attempts that we can predict miscarriages basing only on the images. But there are other ways of predicting miscarriages. You can use ultrasound images. You can use fetal heart videos that you can try to predict miscarriages. So all these things are very exciting that we can really go towards that goal. I will now challenge our clinicians. We know that when you do ovarian reserve assessment and you're looking of antral follicle counts, there are differences between operators and how they measure it and what's the volume, and that will depend also what kind of stimulation protocols we'll do. We need to standardize this protocol. We don't need a manual measuring every day. Computer can do this for us. We just need to find the right way to do that. The same thing is, for example, for uterine lining or trilaminar lining, or even maybe receptivity. All these are projects that we are working currently to see how we can the best standardize the whole procedure. We're actually working on the project that will actually try to review failed IVF cycles and try to identify why the certain cycles failed. And we are trying to use all the knowledge and experience from all of us to try to understand why certain patients fail, and then we can use next cycles, maybe we can be more successful. AI application is already happening. If you are doing donor recipient matching, it's not just about facial recognition or facial matching of features of the patients, but we also need to use genetic profile, demographic rates, and so on. So these things are already in pipeline. So ultimately, we want to actually combine all the parameters that is available to us. We want to combine embryology parameters, clinical parameters, genetic parameters, everything we have. We need to create a big picture, and we need to have AI who can actually assess all this. Humans cannot do this. It's just too much information. This is how I see it. Maybe in the future. We can have AI that will evaluate female and male. We'll try AI to give us proposals, how to stimulate, what's the optimal way to do it. We will have AI who's going to help us how to identify the best embryos in the embryology lab. And then we can eventually look at it. What will be the best cause of action? Should we transfer? Should we do biopsy? Should we cryo? I think all these things needs to be assessed. So in conclusion, for my view, I believe that AI can get us there. I think we need to do a lot of work. There are a lot of questions. We need to do a lot of assessments and studies but I think it could be very good thing that can help us to get there. This is also the first step of automation of IVF lab, and we like it or not. And I'm an embryologist. I work every day in the lab with my hands. This is where we're going to come up to. There are many startup companies, and you will be probably approached of them, and you will see them. I think the bigger question is all these startup companies needs to show us in the peer review matter that their data are validated and they can be used throughout all the United States or country in the world. I would like to thank my group at Cornell, especially Dr. Rozovac, Dr. Elemento, and other people who are participating in all these studies, and also some of the collaborators, outside collaborators. If you want to know more about it or you want to discuss more, I would like to ask you to look at the upcoming fertility and sterility views and reviews that will be actually published in January of next year. And these are the authors, and we will have option to actually look at much more in deep. And I also want to invite everybody to join the new special interest group, AIIVF. We're going to have inaugural meeting this year in 2020, and hopefully I will see you there. Thank you.
Thank you, Dr. Zaninovic. What a wonderful talk. Evelyn, shall we give a couple of questions to Dr. Zaninovic, or shall we go, shall we entertain?
No, that would be great. That would be great. So the first,
go ahead.
we have a question from the panelists. Thank you very much. That was a terrific talk. The first question is, congratulations. Your euploidy and aneuploidy prediction graphs are close enough to argue that clinical application is here. But rather than predicting euploidy, the assay should just classify rank without explicitly mentioning chromosomes. What are your thoughts about this?
Well, I will say, of course, we are looking into, is there a way to do specific chromosomal abnormalities prediction? One of the problems is when you look at your PGT-A reports, you rarely can see single aneuploidies. Like, for example, people are arguing the most frequent is trisomy 21. But if you look at your own data, and we did, we could not find so many examples of the single trisomy 21. It's not so many, because they're always associated with some other abnormalities, or there will be what we call complex abnormal embryos, which has 2 or more abnormalities. So it's going to be very difficult to pinpoint each chromosomal abnormalities, and maybe we don't need to go there. And we know that probably, we actually know that now, that euploid and simple aneuploidy embryos, they look alike. They'll be very difficult to distinguish them just by looking at them. However, I think we are very close to get into the level of looking into the euploidy versus complex abnormal. And I think that's what our goal is going to be.
Nikica, one of our panelists, writes, bias is introduced into all artificial intelligence applications, whatever the field. How can we safeguard against this in assisted reproduction?
Yeah, that's always a problem. As I mentioned before, it's all about how you train your dragon, what they usually call. How you train your computer, what type of data you're putting in. Your data has to be balanced. If you have a lot of negatives and you have a few positives, the computer will learn how to predict negatives more than positives. Now, the other thing is diversity of the data. For example, if I use my algorithms and I move to India, and I want to use my algorithms to Indian IVF people, Indian IVF data, I actually have to use Indian data first, incorporate into the algorithm to predict data from India. That's how I see it. So I think it's going to be very dangerous, and there'll be a lot of startup companies who's going to come out and say, here is the algorithm. It can predict everything you need. But I think we need to be very careful how the data is analyzed, how it's validated. And actually, I will ask people to read these views and reviews, because we are actually have 1 or 2 chapters specifically looking into the how to analyze and what will be the right way of doing all these testings.
indeed, one of our panelists noted that, for instance, you would want to include something environmental like air pollution levels to inform an algorithm locally, yes?
Absolutely. I mean, this is just a scratching the surface. We would like to use as many parameters as we can because we don't really know which parameters will be important. I mean, even with the euploid embryo transfer, you are still having 20 to 30% non-implantation. We need to figure out why and how. What is there that we're missing?
Great. One more question for you. Is software the most important factor for future AI applications?
I will argue that it's not. I mean, right now, you need to have experts in the computer science to create your algorithms. However, more and more we are seeing there is actually big companies like Google, Microsoft, Amazon, they are creating programs that are automatic machine learning programs that you can probably use. It's going to be just a matter of trying them and see how efficient they are. But right now, you still need to have like experts. And that's a little tricky. And you need to merge embryologist with a computer scientist. And maybe it's easier when you are in the academic settings. It's not that simple when you are in the private settings.
Thank you all for your attention this evening. I would like to take a minute to give a big shout out to the founders of I3, Dr. Jacques Cohen, Dr. Peter Naj, Thomas Elliott, Shasta Sarudin, Marianne Svetes, and Giles Palmer. Thank you for the vision and ambition to start this invaluable platform. Additionally, I would like to thank the New England Fertility Society Executive Committee, and in particular, Evelyn Neuber and Michelle Pitcher, who continue to foster our partnership with I3 in order to make our virtual presentations a reality. And thank you to Kathy Doe, the NEFS Vice President, who secured both our speakers for this evening, introduced the speakers, and co-moderated the question and answer sessions. So audience, please join me and give a gigantic virtual applause to the I3 founders and the NEFS Executive Board. Thank you all. I would like to encourage you all again to contact I3 if you would like to help create or submit some website content through their website or at their email, contact@ivfmeeting.com. The group at I3 has been a pleasure to work with for our second virtual presentation of the New England Fertility Society. If you represent a professional organization, please help spread the word and let I3 know if you would like to host a session on your own topic, even in your own language. I3's aim is to continually operate in order to maximize the value to the reproductive healthcare community. Thank you once again to our speakers, Dieter Egley and Nikica Zaninovic, and to the NEFS corporate sponsors, without whom New England Fertility Society would not be able to continue. I would also like to give a big shout out to our audience for showing up at all hours of the day and night, depending on what country you're in, and asking fantastic questions to keep the conversation so stimulating. We appreciate all of your support. A few final reminders. The recording of this meeting will be available on the I3 website shortly. For laboratory staff seeking to obtain credits, you may print out your certificate for this meeting and submit to ABB. For physicians and nurses, you will receive an evaluation by email, or you may go to the New England Fertility Society website, www.nefs.org, and click on the link to complete your virtual meeting evaluation, and then your contact hour certificates will be emailed to you. One final reminder, the 35th I3 session will be held next week on Tuesday, September 29th. The title is COVID Strategies, as in coping strategies during COVID. I3 has asked the IVF community to send in videos of unique ways that they have continued to work through the pandemic. So it'll include videos from around the world and live talks by Brianna Calcavecchia, Dr. Nuno Costa-Borges, and Dr. Debbie Venier. Hopefully you'll be able to attend, and we thank you all for your attention this evening. Have a good night.